Hello, everyone. My name is Dr. Jeremy Petrovich, Developmental Editor for Current Protocols at Wiley. It is my pleasure to introduce our speaker, Dr. Anas Larbi. Dr. Larbi graduated from the University of Sherbrooke in Canada in 2005 at the Clinical Research Center, Faculty of Medicine. He performed his immunological studies at the Research Center on Aging at the Laboratory of Immunity and Signaling. His early work on intracellular signaling events in T cells and neutrophils during aging focused on membrane lipid raft composition and function. He pursued his interest in immunological aging at the Center for Medical Research at the University of Tübingen in Germany, where he focused on the impact of chronic infectious agents such as cytomegalovirus on immunity of the elderly. There he also developed his skills in multi-parametric flow cytometry. He joined the Agency for Science, Technology, and Research in February 2010 to fulfill two missions: to develop a program on immunity in aging humans and to set up a competitive flow cytometry facility in the recently launched Singapore Immunology Network. He has been involved in many educational activities related to cell analysis and in particular flow cytometry. He has published more than 160 papers in the field of immunological analysis with an emphasis on flow cytometry. Dr. Anas Larbi is an emeritus ISAC scholar and involved in many of its activities. With that, I present Dr. Anas Larbi and his webinar titled Making Panel Design Easier in the Era of High-Dimensional Flow Cytometry.
So hello everybody, and thanks for joining this webinar about panel design. So I'm Anas Larbi. I'm a scientist working at the Singapore Immunology Network in Singapore. And today I will cover some aspects of panel design, and one of the take-home messages is that we need some consensus on what to do and how to do it. So we'll propose some core panels, which were designed as the research panels to answer some of the questions we have in the Institute. So I work at the Agency for Science, Technology, and Research, is A*STAR. So A*STAR is a biomedical research institute, and one of the institutes actually is called the Singapore Immunology Network, which is dedicated to the human immunology since 2008. Right from its creation, the institute was built with two pillars: scientific excellence and a unique technological expertise. So at the technological level, the institute was built around several platforms, including the flow cytometry platform, which I set up and I'm still in charge. So there are other platforms we are working with, including the genomics, especially since the emergence of the single-cell analysis, and bioinformatics, due to the increased volume of data, as you know, that we generate on a daily manner. So the platform was recently awarded to be the national platform for flow cytometry for the Singapore ecosystem. And why I'm telling you this? Simply because we need to have a strategy now to increase the reproducibility of the data and the results from different studies, because this is one of the new missions we have now in the context of the national platform. So in this context of a platform, and specifically for the flow cytometry, there are several issues that we have to face daily. And these are in terms of expertise of the staff, operators, or the users, in terms of reproducibility, flexibility when we run flow cytometry experiments. Sometimes we have to stick with some regions we have to use because we started the initial experiments with those regions. How you can compare work that is done across different labs or institutions, and of course to sustain scientific excellence to provide with the scientists with the best for their research. So on top of that, there are additional challenges that are extrinsic ones that could complicate the way each of us may manage a core facility. And this includes the increased variability in the diversity of instruments, protocols, but also the immense number of reagents, fluorescent antibodies, in this case, that are available to the community of cytometrists. So these are not helping a lot to get a consensus, especially for newcomers in the field, on how to perform high-dimension flow cytometry, especially regarding how to design panels. So today, webinar is a lot about how we should move forward, and instead of relying on companies to provide more and more solutions, and us to test which one is the best fit for our, let's say, question, is to at least get a consensus in order to harmonize immunoprofiling, how we should, where we should go, how we should do it. So, of course, there are initiatives and people that are trying to get things improved. And, for instance, you have the Euroflow or the Human Immunophenotyping Consortium in the US that are kind of two driving forces. This is complemented by the ISAC, the International Society for the Advancement of Cytometry, and the affiliated journal, Cytometry Part A. Especially, they publish the OMIP panels. So the OMIP stands for Optimized Multicolor Immunofluorescent Panels. So you will find also in the literature various publications that are looking into those issues. So we are aware that things have to be improved, but we need probably a better, let's say, to be better organized. So, for instance, I have been involved in ISAC and Cyto conferences during which we organize workshops regarding flow cytometry in multi-center and longitudinal studies. And this is particularly of interest to us as we are interested in human aging, and this requires, as you understand, very long studies. So practically, how can I ensure that my flow data from a donor A collected in 2013 will be comparable to the one I'm running in 2015 or 17, 19, or even beyond? Of course, there are many factors that can influence the results, so it's sometimes independent of flow cytometry itself, probably more related to sample collection and preparation. Okay. So to show you how this can be critical, I show you the data generated during a proficiency study we participated a few years ago. The experimental plan was simple. You had 13 labs around the world, sent PPMC from the same donor, as well as tetramers to recognize the CMV-specific CD8 T cells, in this case, to be stained in the sample that was sent. So the test lab had the freedom to choose on the other regions, like the CD8, CD3 antibody, the type of equipment, perform the compensation as you used to do it, and the washing steps. You have a bit of freedom. And I think the data here is quite impressive, and it shows you how the accuracy in the expected frequency is variable. And so you see, even running a basic flow experiment, a 3-color experiment, can still lead to variability. So is there any magic to avoid such issues and improve the way we design panels? Well, there are things we know quite well. The dye properties, the cytometer configuration, the marker density. There are things we know we don't know. Maybe some combination of fluorochrome not tested, especially for the new regions that come up. Possibly the effect of the treatment of the sample, for instance, in the case of tissue dissociation. But this could be known. And of course, there are things we don't expect to happen. But clearly, we know a lot about fluorescence and how the regions we are using react to laser excitation, how neat they are, and which one we can be troublesome, especially for the spectral overlap issues. So with this knowledge, and it's something we know since decades, there is the opportunity actually to see how we could optimize the utilization of this knowledge when it comes to panel design. So we came with the idea of proposing core panels. This idea, of course, is not new per se. As I showed you in the previous slide, some initiatives exist. Today we'll be focusing mostly on the high parameter panel design, although this the same principle applies to maybe less, let's say, extended panels. The issue, for instance, with fixed panels such as in the OMIP, although I have to say they are of great scientific and technical quality, is the lack of flexibility. So the aim of having core panels is to combine the knowledge we have acquired over the years and see how this can be translated into a feasible and long-lasting, as much as possible, strategy, especially in the context of a flow platform where you have many people coming with different ideas, different background, different habits, how you can try to better organize this. So practically, what we've done is we've created core panels by accounting for fluorophores that interact with each other, leaving detectors with spectral overlap available for markers of interest to drop in. So those with less spectral overlap are left free, and this is where the flexibility is interesting, because we know that scientists, they want to have this flexibility to come up with their new marker or in-house antibody and things like that. The idea is to provide them with core panels that will not be too stringent, and they can really come up with and changing actually on the way the markers they drop in. So we tested how the fluorophores in the available detectors affect the core panel. So for instance, how the high marker expression and the spectral overlap affects the core panel, and of course which fluorophore are more suitable for the low expressed marker, because it's also an issue we face daily. Okay. So when we start designing flow cytometry panels, we of course have to keep in mind three pillars that most of us are following. So the spectral overlap. So the core panel attempts actually to take this into account and ensure there is enough separation between the populations. Okay. The stain index, and this is something that we can test each of us in every machine we have, the staining index for each of the channels we have available, and using the corresponding dyes, and to ensure that even if the absolute MFI is low, the stain index and the resolution of population can still be good. And obviously the expression of marker, which is the target of our studies. And we know some can be less expressed versus some others are more highly expressed. And there have been recently some, let's say, studies where there have been, let's say, a systematic analysis of the antigen density, especially in the context of immune cells. So this is something also that we should probably better integrate in the way we design the panel. So we are taking this into consideration. So we considered actually three methods to investigate fluorophore interactions with all the detectors we had. So we were using a Symphony equipment for these experiments. Because, of course, the aim was to analyze the flexibility of the core panel, so we want to look at the suitability of all the remaining channels for high, mid, and low express marker. So to do that, so we selected some markers that follow this low and high, mid level of expression. So CD45, CD4, CD19, CD127. And this is what we've done. So the first method was to physically, as a physical method that we call fluorescence +1. So you know fluorescence, the FMO, fluorescence -1. So here is the fluorescence +1, where you have one tube. In this tube you have cells that are stained with all the antibodies from the core panel, plus one antibody of our choice. and this is what we have acquired. So there are, of course, pros and cons of such a method. The cons is that you have a lot of tubes you have to stain, so using a lot of antibodies if you have to cover a lot of those different single antibodies to be tested. Okay, there are, of course, some apart from the logistic, there are other experimental complications, and this is more related to the competition between epitopes that may affect the MFI, and the co-expression of markers like CD3 and CD4 can complicate the analysis. So the other approach is you could stain actually a large number of cells with one of the core panel actually, and then use the aliquot of the stain cell to be spiked into the single stain cells or even beads. So it works well actually for when you use the CD45 stain beads, but when you use low express marker like CD127, we had some issues actually, because you have increase in the number of negatively stained cells due to the low percentage of CD27 expression in PBMC, for example. So since both the methods have some issues, and I have showed you both previous methods to see how we come to this now, to do what we call digitally mixing, and we took advantage actually of the FlowJo, for instance, to digitally dropping the single stains, and then we combined in a FCS file. So we actually just need to collect one sample that is fully stained with the core panels, and I will present you some of the panels we've tested, and the single stain with the CD45, 4, 19, and 127 in each of the empty colors. And I think this is probably a bit of a heavy experiment, although I think it's really affordable and feasible. But actually, it helps you really to cover the full, let's say, the number of complications you may have if you run a complete set of flow analysis using all the detectors you have in your machine. Okay. So we have done that, and I'm going to present you a little bit more on this now. So this is an example of the digital drop-in method, so in action now, so you can see. So the first plot on the left is the core stain sample, displaying the CD8 expression and how it spills in the APC Cy7 channel. The middle plot and the right plot are CD45 in APC Cy7 and CD127, APC Cy7-2, single stain cells. Okay, so these are just single stain cells. So when the separate files are overlapped in the layout editor on FlowJo, here in the lower left plot, actually, you can see the good resolution of highly expressed marker like CD45 in dark blue, and its effect in the BUV 805 channel in our Symphony machine. However, you see that a lowly expressed marker like CD127, so this is the light blue, will not be ideal due to the low resolution. So now that we have a plan and we know how actually we can improve the design and better understand actually the signal we can detect from the flow machine, we have started, let's say, designing a panel according to our need. So the starting point for designing this panel, we call the whole leukocyte panel, because we wanted first to cover most of immune cells. Okay, so we have data actually, we have mined data we have from the lab in the previous months to better understand the distribution and the diversity of immune cells in blood samples. And these are some of the markers we used to assess the leukocytes. So without going into the details of this panel, so you can see how actually it helps us to resolve quite well a number of immune cells. So the T-SNE here on the left enables you to see the complexity and the clustering, while on the right side you see the various cell types that we could discriminate. We have very prevalent cells like neutrophils, or very rare cells like innate lymphoid cells that we could actually detect very nicely. So the analysis of the markers that help us cluster the population is very important, because this actually enables you to better identify discriminating factors, while you also can identify which maybe markers are redundant. And this is quite important because you can run high dimension flow, but to some extent you are limited by the number of antibodies and marker you can target. So the selection of marker is of course still very important in the design of the panel. So we have published this year a study where we actually performed cell sorting of various cell population, 29 population immune cells actually, and we performed RNA-seq data, and to try to set up a tool to perform deconvolution. So this was the aim of this paper, but it's also for us to enable us to really see how the selection of the marker for the identification of population really end up with the right population, because while it's easy that a CD4 is easily identified as a CD4, we know that in other subsets, sometimes you may have overlap. So as the aim today is to have a consensus, so this is also, there are many ways, other ways probably you can get a consensus of which marker you select. So we have, in the context of this, let's say, core panel design, we have set up three panels, one T cell, one for B and NK, and one for the myeloid cell. And today I will mostly focus on the T cell panel. So our experience with the cell type before starting the panel design, I think it's important to show, because it tells you really that it's good to have experience with the cell of interest, but as a platform we also have requests from scientists to study some cells we don't necessarily have experience with. So we had a good experience in T cells. We do have a lot of collaboration with scientists regarding the myeloid cells, but we are fairly new in the field of B and NK. So it shows you the level of confidence before starting, and eventually how you can still actually run high dimensional flow if you follow this strategy of the digital dropping for the optimization of panel design. So this is just to show you some of the studies we have published in the past years regarding T cells or myeloid cells. So you can really end up doing very high quality flow cytometry in a high dimension at a high dimension level. And today I will show you that there are ways to really improve the way you do it. So of course I showed you a little bit for the let's say how we try to get a consensus on the markers. So these are the markers we have used for our core panels. So the T cell, the B and NK, and the myeloid cell markers we have tested, and this is for human, but the same we have some others for mouse. So I will talk about the T cell core panel now. So these are the markers I showed you in the previous slide, and these are some of the, let's say, reasons why we choose the markers. Of course, some people will say they have a better marker for one population. It's perfectly fine, and we try to have here a consensus based on previous studies and from our own experience. So which fluorochrome you have to tag with which of the marker of interest. So here I show you the configuration of our Symphony analyzers. It's a 5 laser. We have 28 detectors across the 5 laser. We have 5 blue, 7 UV, 3 red, 4 green, and 9 violet detectors. So when you run, when you want to run experiment for a panel design, these are the 3 items, pillars we take into account: spectral overlap, antigen density, and staining index. And really, to optimize the result, you need to find the best consensus between the 3 pillars. You have to decide on which you will compromise and still get an acceptable signal that will not create you too much problem when you want to drop in new markers into a panel. So what is acceptable? So in terms of the spectral overlap, compensation is something we face daily and that we have to take into account. So now most of the acquisition flow cytometers, acquisition softwares are quite good in performing compensation, automatic compensation. Obviously, you have to always look at it because, especially when you run high dimension flow, it can be optimized manually. You have also, you see on the lower left, the spread effect that we observe, and we'll talk about this a bit later. So regarding the staining index, so you see on the left side you see the how you can actually calculate your staining index based on the width of the signal and the spread of the background peak. And you see on the right side some examples of staining index in that were reported for some of the fluorescent the fluorochrome that's been tested. So you can have actually a very good idea of your staining index. You must actually test it for your equipment beforehand to have a better understanding on what you can afford and where you may end up with issues. So the antigen density is very important and is something we all use for the panel design. It's difficult sometimes to predict the density of a protein at the surface or intracellularly because you don't know how patients may regulate it or your activation or in vitro, for instance, how it affects the expression of the marker, upregulation, downregulation. Do you put enough antibody in there to cover to saturate? So it's really something which is very important. So usually what we do is that we use what we call the primary markers we use for the lineage gating. And then we have secondary marker, like more related to activation markers. And you have tertiary, markers, which are the ones that are low expressed. So this is the sequence how we, tag markers to fluorochromes or to select this. But this limits exploration and discovery, I think, because there are ways now, actually, with what I've been introducing to you now, that you can improve that. So there are additional consideration, of course, to improve your flow staining. And this I won't cover too much because we know that. I think it's quite well followed now by the community. So reducing the non-specific binding or antibody titration is very important. The fact of including a cell viability dye. Of course, be aware of your sample processing. I mean, digestion of tissue will affect the expression of your markers. One strategy is also to spread the antigens across different lasers to avoid compensation issues. And I think these are some of the things people are doing quite well now. Take into account your sample type. You know, sometimes we tend to set up our compensations and our, idle experiment on PBMC from a healthy donor. But when you receive the sample from the clinics, it's a different story. Another way actually to expand and increase the capacity of your flow is to use the Sentinel approach to increase the output. So what is the Sentinel approach? So this is something we have been using in the previous years. So practically is the combination of different antibodies into the same detectors. And you can do this if and only if the marker of interest are expressed on different cell types. They are mutually exclusive, and you're sure it's not going to be regulated by activation, for example. So, for example, is to put in detector A, for example, IgD and V delta 2 for gamma delta, IgD for B cells. Put in detector B, IgM and CD8, and you put in detector C, CD3, and detector D, CD19. This enables you to separate with C and D CD3 from the B cells, and then when you drill down, you know that your CD3 don't express IgD and that your B cell don't express the gamma delta receptor. So this is one way. In detector A, you have 2 markers. So it's a 4 detector experiment, but you have 6 markers here. So it's something we have tested. It works quite well. You need, of course, to test that it really works and it does not create problems. So I'm just showing here an example of a Sentinel experiment we have run, where we have CD3 and CD19, and you see, for example, on the top, the IgD and IgM staining in the B cell stain, so upper left. If you add the B and the T stain, so here in the middle is the Sentinel approach, you see that you have the same profile in this case. So it really shows you that adding a CD8 and a V delta 2 in those channels do not affect the signal you detect, because you take the signal out by gating the CD3 out of the B cell stain. And the same actually, if you look at the bottom, on the bottom right, you see a CD8 V delta 2 staining of CD3 cells, and in the middle you see the same staining, but this time you have the IgM and IgD that are added in the staining, and it doesn't affect the level of detection, the detection in terms of intensity of the signal and the frequency of cells you can detect. And you do see in your staining that it's very clean. So really, it's something you can use, and it's just an example here to show you that even if you use those clustering tools, it really helps you actually to separate the signal, and you see actually where your population is standing and where the CD3 is expressed versus the V delta 2, and you see the IgM and IgG is exclusively expressed by the CD19. So it's just a way to validate that it does not impact your work. So practically, the experiment that I'm presenting you now for the T-cell panel, and this is how we run the experiment. So we draw blood and we get PBMC from CPT tubes, and we use 1 million PBMC for each of the staining. Okay. So we run single stain for each of the unused detectors for the 3 core panels. So you see here, these are the unused detectors for the T-cell panel, for the B and NK, and for the myeloid cells. Okay, and those single stains in these empty detectors were dropped in the empty detectors of the panels to analyze the spread and the resolution. So we will focus on the T-cell panel, the T-cell core panel now. So here is the combination of fluorochrome and the markers, and by applying all the principles we know and that many of us are using, plus the one I introduced you today, we actually came up with the core panel. So if you are interested, you can always contact us at a later point, and we can share with you more details about this the panels. But it's really something we have worked out in the previous months to make sure they work fine in your hands. So here I show you the gating strategy and the resolution of the markers, the core panel. And you see very nicely how we can identify the various population, myeloid cells, CD4, CD8, subsets of T cells, gamma delta, the naive memory populations, PD-1 expression, and so on and so on. You see CCR7 on the right, bottom right. You see the expression. We can very nicely discriminate the populations. So here now I'm showing you the spillover of in all the detectors. So the empty channels, they are listed on the right here. They are minimally affected by the existing markers, and you can see, as you can see in the slide here. So that's something which is very important: is how your core panel actually affects your empty channels. Are you going to end up with problems later on? So in this case, it's something we took into account: which marker with which channel, that the empty channels actually remain quite easy for you to use. So this is where our single stain drop-in help to make a theoretical framework to help in the panel design to decide which marker we should be paired with which fluorochrome based on the expression of the marker, antibody brightness, and the compensation. So to do so, we can look at every single stain of a high or low expressed marker across each fluorochrome and observe its effect on every channel. So we show here a bright PE size 7 marker, which is CD45, and how it spreads, for example, in the BB790. That should not affect the core panel, and this is something which we tested across the different core panels, the different combinations of antibodies we've tested. So here I'm just showing you the end result. I'm not showing you everything we have done, where we see actually where issue can come up. So really, we think it's quite of a robust and reproducible panel that we have worked out. And again, we'll be happy to discuss in more detail if you're interested. So in the interest of time, we cannot show you the droppings for every single cell. So what we are showing you here is a scenario where you might wish to add in a high to very high expressed marker, CD4 or CD45-like, to all the empty channels. So we did this because we have to take into account that the compensation spillover from any added marker into the panel. So the resultant graphs here represent what such a full panel may look like with highly expressed marker. Okay. So each of the single stain is represented by a single color, while the core panel is in red. So in red you have your core panel again, and the single stain are in single color, different color. So some fluorochrome like APC size 7, BB515, BUV395, and P size 7 are suitable actually to be paired with the extremely highly marker like CD45. So I think that what we can see from this result is that we actually have come out with a core panel that gives you a very good idea of which highly expressed marker you can use, but also actually in terms of the marker that are lowly expressed, maybe where they can be fit. So here I present you actually the low expressed marker. In this case, we CD127, and others can be used. How they can be integrated in the same core panel that is in red again. So unlike the previous slide to investigate the compensation spillover, which is the problem with highly expressed antigen, the dropping scenario here is to identify how bright fluorophores are suitable for low expressed marker and still give a good resolution. So using 127 as the marker here, we identify the list, and it's on the bottom right, that are suitable for lowly expressed marker. So you see there are still quite a number of channels that can be used. So we have actually collated both forms of droppings into this cheat sheet, which is meant to be a framework for planning an extended panel. So if you will have to use those channels here, which are free, so the core panel are using the other channels, which one you should use for which level of expression of the marker. So you can use this really as a way to design your panel. So of course there are still limitations when you run such experiments, and actually it depends a lot on the configuration of your cytometer. So what we are showing you today, although it's on one equipment, Symphony, each of the equipment have their slight, let's say, differences. So you need to really test this again based on your filter configuration, the power of your laser, quality of PMTs. Is it something you're doing on blood? Or, I mean, we did it on blood, so we have not optimized it necessarily for tissue. The availability of reagent is a limitation, but this is one of the advantages of the panel we designed, the core panel we designed, is that we leave the free, the empty channels for regions that are easily found by many of the antibody providers. So it's an advantage here. So, and of course, we depends on the fluorochrome you use in the corresponding channel. Yeah. So, for instance, we use APC, but you will see that if you use a like dye, you will have to run again this type of analysis and comparison. But one way actually to really go around this, let's say, especially regarding the cytometers equipment-specific difference, is to calculate your spillover spreading matrix, because the sensitivity of the detector is affected by the intensity of the signal spillover, and the error actually is largely due to the photon detection, and this is what we know as the Poisson error, which is proportional to the square foot of intensity of the signal. So this is the theory, but it's a very good indicator of the instrument performance, and you can go back to this original reference from Nguyen et al., so it's Mario Rederer's team, where they published some years ago already how you calculate it, and where actually you can improve even your panel design based on your the performance of your equipment. So I'm actually presenting you here the T-cell core panel spillover spreading matrix, and you see the value are very much acceptable, and it shows you actually in the event you have issues or values that are high, so over than 6 probably, this is where you may end up with issues later on when you test your panel. So we know that the core panel works quite well, so we were able to resolve the main populations. So we want now to extend it to the T-cell helper family and also intracellular staining. So we have a core panel of many markers, and imagine now that you want to use this core panel and extend it with the empty channels. This is what we're going to show you now. So we start with the core panels plus the T helper markers. So these are the markers on the left side. So this is the core panel, and you see on the right side some of the markers we have added to this core panel in order to achieve the detection of some of the subsets as shown in the slide. So here I'm showing you the gating of the populations of the extended T-cell core panel. So this is quite of a full panel using the full capacity of the Symphony, and you see the nice resolution of the populations. Here I show you the CD8 cells, and here in this slide I show you how the CD4 subsets actually look like and the different populations of T helper cells. You see the Th2 with the CrTH2, for example, staining here in the middle of the slide. So this is a marker, CrTH2 we added as an additional marker. So to summarize a bit the addition of the markers, so on the left side you have the phenograph of so you see in the heat map the different cluster we are able to identify with the TSNE, the clustering method. And on the right side is the same core panel to which we have added additional markers. So you see you have, of course, more clusters due to more markers you add in. And this is quite, I don't go into detail here. If you're interested, we can follow the conversation. So, of course, we are used to see how the compensation is. So I've shown you in the previous slide the spillover spreading matrix, so that was acceptable. Now I'm showing you the compensation matrix, and most of the values here are acceptable, although the absolute value doesn't mean much, but it's just to show you don't end up with major problems. If you use this principle of the core panel, how you design it, the dropping to better understand your machine, better understand the spillover, and to optimize the panel design, then you end up with 25 plus markers without having issues with compensation. So now I'll show you the same core panels, but to which we have added, instead of surface marker for TH population, is for intracellular staining. And in this slide, I'm showing you the stains we have added. So we have transcription factors, so FOXP3, AOMS, Tibet. We also have granzyme B related to cytotoxic activity of the cells. We also have added some other markers to show, like adding extracellular and intracellular additional markers. So here you see the gating after the intracellular staining, and clearly, and we'll just stick to the lower plots where you see the Tibet and AOMS. You can nicely resolve them. The CD57 with the granzyme B, you see there. The raw gamma T, you see the few population expressing this, and also the FOXP3 staining. Okay. So basically, this is just to show you that, again, same as for the TH marker, adding additional marker in a well-designed panel enables you to immediately end up with a nice result. We didn't have to run a series of testing and changing the combinations to end up with this result. By keep spending time on the thinking enables you to optimize also in terms of saving time and cost and ending up with such a stain. So of course you always have this last-minute experiment, this guy coming to the facility and asking you, oh, can you really help me? I'm struggling. I have this sample coming tomorrow from the clinic, and I need to study this, and this marker. So we try this scenario. I ask Yoi, who is the one that is running some of this experiment in my lab, can you just try to dump a series of antibody in the free channels? And don't care about which marker it is, more or less, just for us to see really how you can do this, how you perform in this scenario, where you don't have so much time to think about the additional markers. A bit to test a bit how robust the core panel is and flexible it is. So this is an example of a 28-color panel based on the T-cell core panel on the left side, you see. And on the right side, these are the additional markers we tested. Some of the markers, actually, we didn't know what we should expect in terms of expression. So here I'm showing the gating strategy and the staining we have applied. And actually, we are quite happy, really, about the results we had in the end. We could very nicely resolve many of the population of interest. You see in the middle CD56, TIGIT 6, 3CR1, TIM3, PD1, and many of the other markers we have added. And this is just to summarize, based on this in this experiment, this trial experiment, the different populations we were able to identify using those additional markers. So really, it's to show you that it's a very flexible. We came up with a flexible core panel that enables to fit various scenarios. We also have worked on a B and NK cell core panel. We won't have time today to cover the same level of detail. So I'm just showing you for this B and NK panel how we are able to resolve the different populations of B and NK cells, and using the IGs like IgM and IgD and 27, you can really nicely see the populations you see on the lower left plots, while we could also identify the subset of NK cells on the top right plot. So all markers and population were resolved. So we also have worked on the myeloid core panel, and same as for the B cells, we'll cover mostly the basics. So here I show you actually the how we are able to resolve the different markers and populations as rare as the PDCs and others. So we have the neutrophils, eosinophils can also be identified in whole blood, obviously. And for the rest, in the PBMC, you see the different cells, basophils, DC subsets, monocyte subsets, and so on and so on. So again, I won't go into details for this. If you're interested by these core panels and some of the details, we can reach out. So clearly, today's presentation was to show you actually how we have conceptualized and the framework for panel design, the core panel design. We think that we have very good quality in terms of resolution of the main population of interest, and we have worked out the core panels in a way that most of the problems actually are solved already, because this is what we took care of. And what you have left with is a series of channels that are free for you to drop in your markers. So we are guided by basics of flow cytometry, staining index, spectral overlap, and antigen density. But we spend time and working with the team, actually the BD team in US and in Singapore to make this let's say optimize and work it out faster. So there are a few people I'd like to acknowledge that helped in this work. So Yoi, so you see his face here, has been working a lot on this the design of the core panels, as well as the other team members. The flow facility in the Singapore Immunology Network for providing us with impressive and fantastic support and it's been funded by some of the grants that are mentioned here. The International Society for Advancement of Cytometry, so ISAC, so I'm emeritus ISAC scholar, so it's really great experience being working with the experts in the world. And last but not least, the BD Bioscience team, they've been very helpful with a lot of iterations, it's been a great collaboration, especially thanks to Bob Alderas, been supporting this work very well, and I think it's been quite of a learning from both sides, and I think it's really important to highlight the work working with reagent and equipment providers, because I think it's a win-win situation where we improve the knowledge and they better understand our need, and then they can accommodate better research. And this time is interesting because, as I said in one of the first slides, this consensus is how we can actually improve the type, not just the amount, but the type of reagents that are offered to us to do our research. Finally, I thank you for your attention and attending this webinar. I hope it was useful for you. Again, the aim of these core panels is how we can work better and work together. So we'll be happy to get your feedback, and we are very open to share with you more information. So thank you very much.
Thank you, Dr. Larby, for that exciting presentation. Our first question from the audience is, can you please highlight the importance of using a viability dye in these high parameter panels?
Yes, very good question, as this is something which is very important to clean the signal we are detecting. So very often we work with either the cryopreserved samples or samples that need some treatment in the case of tissue dissociation. So you clearly need a viability dye to exclude your dead cells from the signal you analyze. So there are a lot of viability dyes actually that are available now, and some of them actually the fluorescence is also preserved after fixation. So these are more the protein binding dyes, and this is quite important, especially for intracellular staining, as often your positive cells, in the case of a cytokine analysis, they may just represent a tiny fraction of the whole population. So it's not unusual to have 5 or 10 or 15% of dead events, especially when the freeze-thaw process is not so well mastered, or when the dissociation process is quite harsh. Sometimes you need it for some tissue. So by excluding the dead cells, it's something you avoid, such as the unspecific binding of antibodies, and this will result in the false positive signal which you don't want, or to some autofluorescence. So really, it's important to get rid of those signals that may actually hinder the signal you wish to detect.
Thank you. Second question is the drop-in cheat sheet based on the properties, primarily brightness, of each fluorochrome?
Yeah, so actually the drop-in cheat sheet that I showed you, yes, so it's the result of the experience we have combining the fluorochromes and the markers that are either highly expressed. So we use the CD4 and CD127 as a model here. So the cheat sheet summarizes which fluorochrome you can use with a low express marker, so that's CD127, and which one can be used with a highly expressed marker. So yes, obviously it's a combination of staining index and antigen density.
Thank you. Next, how do you measure instrument stain index and spillover?
Okay, so the staining index is a way actually you measure the brightness, the relative brightness of the fluorochrome. And it's very useful actually for ranking which one are brighter, especially in your instrument. So it's a relative number. So it's better to focus on the not on the absolute value, but how the different fluorochrome staining index is higher or lower. So you can obviously find some websites actually that can give you a relative ranking of say the brightness using standard analysis. I think they are more or less all let's say matching, but it's still important that you do it yourself in your instrument. There are also existing tools. I mean, if you follow the work from Pratip at NYU, you have the color wheel, which is a tool for the design of panels also, which integrates this aspect of staining index. So if you want to do this, actually, you can use actually the same clone for a high density marker, for example, which will be conjugated for with each of the fluorochrome you have in your that can be detected in your equipment. For example, you have the BD, they have a kit with a CD4 clone, or you can buy the antibody individually. That can help you to make this in a systematic manner.
Thank you. What colors were used for the extra TH and ICC markers?
Okay, so for these two extra, let's say additional markers, we actually have used mostly the same fluorochromes with different antibodies. So actually it's really based on the cheat sheet that I showed you. At this stage, when you have the cheat sheet in terms of which marker, in terms of the density of the marker and which fluorochrome you can combine, it's not really important anymore which fluorochrome you have used because the data in theory should be quite robust based on the assessment we have done and some of the dropping data I showed you in some of the slides, the same actually has applied to the for the B cell and the NK and the myeloid that I didn't have time to show you, but I really encourage you to contact me to look further in these panels.
Thank you. So, given that there is such a high density of new biomarkers that can be tested nowadays at once, how can a user be certain that the fluorescent level of the biomarkers tested is a real positive and not just due to high background?
Yeah, obviously, when you end up with so many markers, you want to ensure that it's something that it's a signal that is real. And we have seen in the literature and some people reporting some papers, although in those high impact journals, where it's sometimes true that you can debate about the quality of the signal that is detected. Is it a real positive? Is it because of compensation? Is it because of high background? So I think the best that can be done is still the fluorescence -1, so the FMO, that which actually consists of running your whole panel except the marker for which you have a doubt. We are running here in our platform a clinical study for a pharma where they request to run the FMO for each of the markers for each of the patients. So you see it can go very intense in terms of the workload, but this is one way actually, and I think it's the best way to ensure that your signal is real positive.
Thank you. We've reached the end of our time today for questions. I'd like to thank Dr. Anis Larbi for taking the time to present to us his talk on panel design for flow cytometry. Any questions that were not addressed will be answered by email. This presentation was sponsored by BD Biosciences in partnership with Wiley's Current Protocols. This webinar will be available for on-demand viewing on the Current Protocols website at www.currentprotocols.com in the webinar section. Thank you for attending.